I really would like to thank the students for inviting me to present our research at this conference. And I am a professor in the Department of Biology at Penn State University in the United States. But this year, I am on sabbatical at ISEM at the University of Montpellier. And if you have any questions after my talk, here is my email. Feel free to send me an email afterwards. So I know that the topic today is omics. So I will be talking about omics data in the case of childhood obesity. So as disease-related omics data. So a little bit more about my lab and about my research. So we work on 4 different topics in the lab. One is on the sex chromosome evolution in vertebrates. Another one is on the mitochondrial DNA evolution in mammals. And on the one more topic, and this is what I work on largely here as part of my sabbatical, is on non-B DNA and evolution. And today I'm going to talk about the childhood obesity. I already gave a talk last semester on sex chromosome evolution in vertebrates. And I'm planning on giving a talk about the mitochondrial DNA evolution in mammals so that the scientific community in Montpellier will be familiar with what my lab is doing in detail. But back to omics. So what is omics? Omics is the analysis of a large variety of biological data. I will start with a brief description of a study that was recently completed. According to one research study, it was really interesting to investigate the effects of traveling to space on the human body. And the experiment that was conducted is that 2 twins, Scott Kelly and Mark Kelly. One of them actually went to space in 2018 and was there for about a year, while his brother, Mark Kelly, stayed on the Earth and was enjoying his retirement. And during that time, and particularly after this time, a variety of biological information types were compared between these 2 identical twins. In particular, what was compared is their genomes. Well, they are almost completely identical because these are the identical twins. But genomics is the type of omics that actually gathers the information about an individual's genome, frequently at the single cell level. Another type of information that was studied is epigenomics, and these are the modifications of DNA, frequently with methyl groups, that affect the gene expression, that affect the other type of omics, which is called transcriptomics. And transcriptomics is the analysis of the data on gene expression, of all the genes that are being expressed by a cell, by an organism, including different expression levels and so on. The next level of omics that was studied is the proteomics. And this is the collection of proteins that are being expressed by the cell or by the organism. And another really interesting type of omics is the microbiomics. Microbiomics is the type of omics that represents the collection of all the microorganisms in the human body, including the beneficial bacteria in the human gut, but also many bacteria that are present on our skin. It is really informative to study microbiomics to understand health and disease. And another type of omics that I will be referring to is actually metabolomics. And metabolomics is the type of omics, the type of data that investigates the metabolites. And the metabolites are frequently the consequences, the results of reactions, cellular reactions, both by the body of the host, the human, but additionally by our microbes that live in the human body. So when we talk about metabolomics, both the host and the microbes are contributing to this collection of microbes. So going back very quickly to the study of the twins, one of them went to space, they were found some interesting differences which are related to humans going to space. First of all, there is an increased level of radiation in space. So some results were found at the level of genomes. And the different telomere length was found between the person who traveled to space as compared to the one who didn't travel to space. And this was related to stress as one of potential explanations. There was also differences in the levels of gene expression, also likely related to stress in space, but also to the activation of immune system. The microbiome in space actually lost some diversity. So there is, in space, there is a rather clean environment compared to what we have on Earth. So I'm going to give you an opportunity to take a quiz, maybe at your own time, if you would like to revisit what different types of omics are. Here is a quiz, and we're not going to take it now, but if you want to do this at your own time, you can take a screenshot and do it at your leisure. And here, each of the omics types that I was talking about is explained. You just need to match the correct omics with the correct definition. And if you're not sure about the correct answers, here I'm also giving you the correct answers. But just to give you one definition to remember, and it's omics itself. It is an integrative field of study that helps scientists determine what each part of someone's genetic instruction does, how it relates to the other parts, and how each part is expressed. Then after all of this data are collected, different types of omics data are collected, this frequently very large and complex data sets undergo substantial analysis. And this is where we're using statistical approaches, machine learning approaches, deep learning, and we integrate the data from omics, from different types of omics, into what we call multi-omics, transomics. We integrate different omics data types. And this integration allows us to build models that can predict health and disease, and that can affect the phenotype. So today I'm going to talk about our study of omics approaches as related to childhood obesity. And this is something that my lab has been working on now for about 10 years. So why are we interested in childhood obesity? childhood obesity is the very important problem of our times, in particular in the United States. About 17% of kids of the age of 6 to 11 years are overweight or obese. And 70% of kids who are obese are going to be obese as adults. So it is really a huge problem. And childhood obesity has many complications. Frequently, these are psychosocial complications. These kids have low self-esteem, depression, low quality of life, the sense of low quality of life, but also more physiological. Many cardiovascular, endocrine problems, gastrointestinal problems, and musculoskeletal problems as well. So we wanted to really contribute to understanding what is causing this disease. And the causes of childhood obesity are multiple. Some are unmodifiable, including genetics. So there are a number of genes, many of them are probably still undiscovered, that contribute to childhood obesity. And this is something that is impossible to change at this point. But then there are many modifiable factors as well. This includes physical activity, diet, sleep. The quality and duration of sleep can affect obesity. Also, parental determinants such as smoking, working schedule. There are also many intrauterine factors, and here they are called unmodifiable, but I would argue that some of them can be modifiable if there is substantial education, but some are not. For example, gestational diabetes, this is something that cannot be modified, while education on gaining weight during pregnancy is somewhat modifiable, although not completely. And epigenetics, these modifications of DNA can frequently be an important factor in affecting childhood obesity during the intrauterine growth. So I was fortunate to be part of a very interesting study conducted at Penn State at our School of Medicine. This study is called INSIGHT, Intervention Nurses Start Infants Growing on Healthy Trajectories. And in this study, 279 families were involved. And at the beginning, only their firstborn children were part of the study. But then also the second-born child from these families was involved as well. So the siblings, and this second study was called SIP-SIGHT. And so what was collected for these children? What kind of data and what kind of samples were collected? We collected data on the mode of delivery, whether this was a vaginal birth or cesarean section. Then the weight and length or height were collected at multiple doctor visits for these children. Also, we had information about maternal weight, height, and health. The same for the father. A lot of information about baby health, about baby medication. Also, very detailed information about baby diet, whether the baby was bottle-fed or fed with breast milk. And then when the solid foods were introduced, then we collected blood from a finger stick from the child at birth, but also at the age of 1 year. And we collected multiple samples from the microbiome. And in this case, we were collecting the samples from the gut microbiome, meaning from stool, and also from the oral cavity. We also collected placenta and cord blood from these children at the time of birth. And this is a very large study which included several collaborators, in particular Ian Paul, who is a pediatrician, who actually is interested in the social aspects of childhood obesity. So he is teaching parents to recognize hunger cues, to really see whether the child is crying because the child is hungry, or perhaps the child is sick, or perhaps the child wants to have some interaction with a parent, or maybe the child just needs some attention. But you can imagine how much data were collected with this study. So I really needed help from statisticians, and 2 statisticians were on the team for this study. And also several graduate students and postdocs, Sarah and Anna and Dipmalia. So all of them contributed to this study. So what kind of omics we were studying? We were collecting the data and studying the data on 4 types of omics, and these are all at different stages of completion. We were studying microbiomics, and this study is published. Metabolomics, this is also published. Genomics, this is currently in press. And epigenomics, and this is an ongoing effort. So I'm going to talk about each of these studies so that you could understand the omics signatures we found of childhood obesity in the first 2-3 years after birth. And I will start with the microbiome. And the question that we asked here is whether there is an association between microbiome and obesity in young children. And healthy microbiome is diverse. So the microbiome of a lean person has a greater number of different species of bacteria than a microbiome of an obese person. So our microbiome, especially in the gut, depends a lot on the diet that we have. So healthy diet frequently means a healthy microbiome. But then the microbes release the metabolites that stimulate the production of hormones by the gut. And this hormones then send the signals to the brain and to other organs to regulate the metabolism. So this is how the gut microbiome is thought to regulate obesity. And you can imagine that any problems, any misregulation of the system can lead to diseases. And in fact, this are not just the metabolic diseases, obesity and diabetes, but also heart disease, neurological diseases, and even diseases related to immune system and allergies have all been linked to the function of microbiome. How is the microbiome of a child is established? The first gift that the mother gives to her child is the gift of microbiome. And in fact, the microbiome is different for the children that are born vaginally versus the children who undergo a cesarean delivery. And the next contribution to the microbiome of a baby is of food. There is a difference in the microbiome of the children depending on whether they are breastfed or whether they consume the artificial milk from the bottle. Also, if the child takes antibiotics, his or her microbiome will be affected. The environment will contribute significantly to the microbiome. For example, if there is a pet in the house, this will affect the microbiome. But at the age of around 2 or 3 years, the child will have likely an adult-like microbiome, particularly because the foods that the child is eating are largely the same as the adult foods. So the study I'm going to talk about has been published. So this is our publication from 2018 in Scientific Reports. And the question that we wanted to ask is whether there is a relationship between growth and the gut microbiome in infants, and whether there is a relationship between growth and the oral microbiome in infants. And we analyzed 226 mother-child pairs for which we collected oral and gut microbiomes, and we built child growth trajectories. So here we used advanced statistical analysis technique called the functional data analysis, because we built functions of child growth. So here on the y-axis is the growth index. So it's weight divided by height. And we had multiple time points. So we built a function of how the growth index changes across time from birth until the age of 2 years. And this is just for one child on the left, and this is for multiple children on the right. So there is quite a variety. And we wanted to see whether there is a relationship between this growth index and the composition of the microbiome of the children. So what we found is that the oral microbiome, so the microbiome of the oral cavity of 2-year-old children, is associated with those growth functions, with growth trajectories. And it is associated with rapid infant weight gain. On the other hand, the gut microbiome of 2-year-old children is not associated with rapid infant weight gain. It's not associated with growth trajectories, but it is influenced by diet. So it seems like the signatures of obesity may be established earlier in oral microbiome than in gut microbiome. And this suggests that the clinical test can be developed, that if we sample the microbiome of a child, we have predictive power of saying whether this child will develop obesity or not. So to summarize this part of the talk, is there an association between microbiome and obesity in young children? Yes, the answer is yes, particularly for oral microbiome. So let me move on. And we next wanted to investigate whether there is an association between metabolites, between the metabolomics and obesity in young children. And this work was done in collaboration with Andrew Patterson and was recently published in Pediatric Obesity. So here, we really are interested how the gut metabolites are influencing the child weight outcomes. And we also are considering the maternal health and habits during pregnancy, delivery mode, but also child's diet, antibiotic usage, and so on. But when we talk about the gut metabolites, we frequently think about the metabolites that are contributed by the host, but also that are contributed by the microbes of the gut. So here we did the nuclear magnetic resonance analysis of 189 stool samples for the firstborn children that we recruited for the microbiome study. And we found one particular metabolite that correlates significantly with the BMI, with the growth index. So there was a positive association between butyrate and body mass index. And we also found that children who were obese or overweight had a higher butyrate metabolic concentration. We wanted to find out what are the particular bacteria that are synthesizing butyrate in our sample. We found that the butyrate concentration was correlated with the abundance of butyrate producing bacteria, in particular of butyrate producing firmicute bacteria, and one particular genus of bacteria, the Fecalibacterium, correlated the best with the abundance of butyrate. So what is really going on? So this particular metabolite, the butyrate, results from anaerobic microbial fermentation of non-digestive carbohydrates such as starch, dietary fiber, and so on in the host intestine and colon. And it influences host physiology by signaling through fatty acid receptors and the associated hormones. So when activated, these hormones can affect appetite and can influence the sense of fullness. So this is how we believe butyrate works to affect childhood obesity. So to summarize this part of the talk, we were able to show an association between metabolites and obesity in young children. Let me move forward and talk about genomics. We wanted to ask a question whether there is an association between genomic variants and obesity in young children. And this is a paper that is available online. It's currently in press. It's available on bioRxiv, in which we constructed a polygenic risk score for childhood obesity. And the previous genome-wide associating studies studied adult obesity. They used huge sample sizes, but frequently only one data point, such as BMI at a certain age. What we did here is we investigated childhood obesity, and we used a small sample size, only 226 individuals, but we used multiple data points on a growth curve using the functional data analysis. Because we used multiple data points, our power increased by 5-fold. So studying 226 individuals was the same as studying 1,000 individuals. And what we found is that 24 single nucleotide polymorphisms, or SNPs, can predict childhood obesity really well. We even narrowed this down to only 5 SNPs that are related to childhood obesity. They can predict childhood obesity in other cohorts. They also can predict obesity in adult cohorts. And this is very important because when we're using SNPs from adult cohorts, we cannot explain childhood obesity. So potentially the genetics of childhood and adult obesity is not exactly the same. And we can identify kids who are at most risk for childhood obesity because of their genetics. And these are the kids that can benefit most from the intervention. So to answer the question, is there an association between genomic variants and obesity in young children? Yes, definitely there is an association between them. So the study on the epigenome is still ongoing. So we want to ask a question whether there is an association between epigenetic modifications and obesity in young children. For this, we've collected cord blood samples and placenta samples from the same cohort. And this is a study that is being led by Sarah Craig and Delphine Lariviere. By the way, Delphine did her PhD at the University of Montpellier. But I don't have any answers yet. I suspect that the answer will be yes, but I don't want to jump ahead of myself. So just to conclude, what we found is that there are strong effects of microbiome on childhood obesity and also strong effects of genetics. There are also some subtle effects of metabolites. In particular, we found one highly significant metabolites. And we still need to determine the epigenomic effects. And eventually, we would like to build an integrative model of childhood obesity using all these different types of omics data that we have collected. So please stay tuned for more developments of this topic. And I would like to acknowledge all the individuals and also our sources of funding. And I would like to end with a picture of my lab from one of the last Halloween parties we had a couple of years ago.